everyone, welcome to Botany Insider. So in this particular lecture, we'll be dealing with one of the most important topic from the CSIR point of view, and along with that for the DBT exam and any other life sciences exam stream that you are preparing for. Reason being, this is most frequently repeated and also very important topic, and that is cancer. In this particular lecture, we'll be dealing with that what cancer is all about and also about the various genes involved. That is, we'll try to have a complete overview for the cancer biology. So let's get into the video now. So first of all, we need to understand about that what cancer is all about. Reason being, we tend or we use this particular word a lot of time in the day-to-day life, or you hear about this in the news or anywhere else. So you need to have a clear understanding about that what cancer is all about. So if we want to define cancer, it is the uncontrolled growth of the cell, of the abnormal cells anywhere in the body. That means, let's consider this is a cell. So let's take an example for the same, and that is, let's consider two conditions. The first condition is that we have a normal cell. This is a normal cell and this is a cancerous cell, let's say. So in case of normal cell, what happens is, every cell have a specific lifespan. That is, they have a particular number of days or particular number of time period for which they would be alive in the cell and complete off with their function. That is, we have this normal cell, and this normal cell is completing their function, or it is completing of all the metabolic activities which it was assigned for. So let's say at this particular stage, all of its activity got completed, and now this particular cell have completed the functions. Now it want to die off. Reason being, there is no additional work that it have to do and its life is now over. So now this particular cell would die off, and the process by which it would die off is what is known as the apoptosis. We have already dealt about apoptosis process in complete detail on the channel itself in 2 parts. I'll provide the link of the same in the description box. Check that out because that is also a very important topic. So do not skip that topic. So this was about a normal cell life. Now let's consider the second condition in which there occur the cancer, or we can say that this cell has turned into a cancerous cell from a normal cell. Now what happens is that in the cancerous cell, this cell would continue to divide irrespective of any function, or irrespective of that what its role is. It would continue to divide and increase in number without having any stoppage. That is, the cancerous cell, or in the cancerous cell, there is no property or there is no phenomenon of apoptosis, and because of that there occurs an uncontrolled growth, and this uncontrolled growth results in the formation of a mass of cell being accumulated in any part of the body. This cancer could occur in any part of the body depending upon the origin of that particular cell, or depending upon that in which part of the body which cell have converted into the cancerous cell. So in very simple language, if you want to have a simple definition for cancer, it would be that cancer is the uncontrolled growth of the abnormal cells anywhere in the body. A disease in which the abnormal cells divide uncontrollably and destroy the body tissue, as we have just discussed, and these abnormal cells are termed as the cancer cell, malignant cells, or the tumor cells. So we can term, or we can name these abnormal cells as cancer cells, or malignant cells, or the tumor cells. Now what happens is that as a result of the cancer, there occurs the formation of the tumors, and the most lethal or the most death causing activity or the most death causing property of these cancer cells is metastasis. Now what is metastasis? Let's understand this also. So in very simple words, let's consider that we have this particular cell, and this cell is now a cancerous cell. It is dividing uncontrollably and its number is getting increased without having any stoppage. There is no apoptosis going on, and this cell is completely dividing into as many numbers as possible. Consider that this particular organism was having the cancer originated in the throat area. Now what happens is that these cancerous cells or these malignant cells, there occurs or they possesses a property of metastasis. And what is metastasis? So metastasis means that when any of a cell from this particular location detaches from this particular area and goes to any new location and cause cancer there. So the property of traveling, in simple or in very lame words if you want to define it, so the property of the cancer or the ability of the cancer cell to detach from one area and invade into any new location and cause cancer there, this is what is metastasis all about. So as you can see that what metastasis is, that it is a lethal property. What happens in this condition is that there occurs the spread of the tumor within the body and there occurs the production of the secondary tumors in the new location of the body. So this was about the metastasis. Now let's understand about the basic properties of cancer. We have just discussed about this and we'll just quickly have the list of the same. The first one is the loss of the growth control, that is they continue to grow in the absence of the stimulatory signals. There is no requirement of these cells in the body but the cell continue to divide. Second one is immortal, that is they divide indefinitely, that means there occurs no death of these cells, and the reason for the same is that there occurs the presence of telomerase in the cancer cell. Now what is telomerase in the first go? So if you remember about the genetics lecture, we have discussed about this, that is telomerase is an enzyme which is present at the ends of the chromosome. So because of this particular chromosome, what happens is that it helps or this enzyme helps to maintain the telomeres at the end of the chromosome, thus allowing the cell to continue to divide. So if the telomerase is present in any cell, that particular cell will have the ability to divide. But in a normal cell, after a certain point of time, telomerase enzyme diminishes and that cell dies off. But in case of the cancer cell, this telomerase enzyme is present all through and the cell continue to divide and produce the tumor in the body. So we just understood about this, that we have normal cell. In normal cell, in the presence of the growth factors, the cell would divide, but in the absence of the growth factor, there is no division. But in case of cancer cell, irrespective of presence or absence of the growth factors, the cell would divide. This was about the normal or the basic properties of the cell or the cancer cells. So as you can see, we have discussed about this. नॉर्मल सेल डिवीजन एपोप्टोसिस होकर वो सेल डेथ हो जाएगा, बट इन केस ऑफ द कैंसर सेल this cell continues to divide and form tumors and do not die off. In the healthy cells there occurs the death, but in case of the tumor cells, which is immortal and would do not die off at any particular point and would continue to divide all through its life. Now there are two growths of cancer. The very first is the primary growth and the second one is the secondary growth. The primary growth is the one which have a neoplastic growth or the original growth, or we have it is present in the original location. The second one is the secondary growth, which is the metastasis or which occurs because of the metastasis at different locations. So as you can see in the figure, this is from my notes itself. We have the cell at the beginning, there occurs mutation or we have the mutated cell. Initially what happens is that there occurs the benign growth. So now we have two types of simple cancer if you want to define. The first one is the benign and the second one is the malignant. So benign is the one which remains at the same position and do not spread off, but malignant is the one which grows and which detaches from the normal location or the original location and switch overs to the new part also. So this was about the benign growth. So benign growth is localized. Now it leads to the release of some lytic factors, which is the breaking down factors, because of which there occurs invasiveness and it becomes larger in size, and as a result of that, because of the malignancy, there occurs the spreading of these large sized into the new location, and there occurs metastasis and then the secondary growth. So now there is one more term, and that is angiogenesis. What is angiogenesis? So if the cell is dividing, it would also require resources, or it would also require nutrition to grow off, and for the cell the most important nutritional source would be the blood, and for blood there would occur, or there would be the requirement of the capillaries, so or the vessels. So the formation or the process of formation of the blood vessels is what is given the name of angiogenesis. So this is the simple diagram, or this is a simple process by which the cancer occurs. So now we have the tumor cells, in which first one is the primary growth, and in case of the primary growth we have these many properties. The very first one is that there occurs cell to cell adhesion being present. The cell to ECM, that is extracellular matrix adhesion, is also present. Third one is that the cell is localized, there is no invasiveness, they do not release the lytic factors, they divide like the normal cells, but the apoptosis is absent. Absence of apoptosis is the first and the most important characteristic features of the cancer cell that we are talking about. Next one is about the secondary growth or the secondary cells. So now there occurs increase in the growth cycle, there occurs decrease in the doubling time, that is the cell starts to divide at a much faster rate. Third one is that there occurs the lytic factor release like the metalloproteases and also the collagenase. There occurs damage of the basement layer, invasiveness develops, they become isolated, contact inhibition is lost, and also there occurs metastasis. What is contact inhibition? So when two cells are in close proximity to each other, they tend to remain together, but in case of the secondary growth of the cancer cells, the contact inhibition is lost, and they become isolated from each other, and hence they move to the new location by the process of metastasis and cause cancer there. So this was about it. Now we have the properties of the malignant cells. The very first property of the malignant cell is that there is a enlarged nucleus, prominent nucleolus is present, there occurs increase in the nuclear pores size and also the number. Then we have the cytoplasm turning into the pale color, the Golgi or the Golgi body is poorly developed, and also there occurs lack of differentiation. So this was about it. Now we have some of the genes involved in the cancer, and the two genes or the two most common genes that are involved are, the first one are the oncogenes and the second one are the tumor suppressor genes or the TS gene. So let's understand about each one of them. So the very first one is the oncogenes. So we have to remember about these very nicely, reason being एग्जाम के अंदर इस एरिया से बहुत बार क्वेश्चंस आते हैं, so it is very important. So in oncogenes only one mutation is sufficient to cause the cancer. Remember about this line very nicely because it is very important, and also from this area itself questions are framed. The mutated oncogene is the dominant one and initially it is inactive, that is initially we have the proto-oncogenes. So proto-oncogenes have the gain of function mutation, and because of which it converts into oncogenes. That is initially we have the oncogenes present in the form of proto-oncogenes and they are inactive in nature. But when there occurs a mutation in them, and there occurs single mutation in them because the mutated oncogene is dominant, and we have learnt about this, that in case of the dominant gene only single gene is sufficient to cause the end product or the end result. So the proto-oncogenes initially being inactive, there occurs gain of function and then they convert into the oncogenes. So this particular gain of function could be either because of the point mutation, because of gene amplification, gene deletion, or translocation and the transposons. And the most common examples for the oncogenes include RTK, RAS-MAP kinase pathway, then we have JAK-STAT pathway and the serine threonine kinase pathway. We will be dealing about each one of them when we will be studying about the cell signaling. For the time being, just remember about these examples. Now the second one is the tumor suppressor genes. So tumor suppressor genes require two mutations. There occurs, or there is the recessive mutation in both of the alleles being required. There occurs loss of function, and the examples are P53, RB and PTN. So now this was about the oncogenes and the tumor suppressor genes. Let's have a quick review or the quick overview for the same. So we have the genes being involved in the case of the cancer. The very first genes are the oncogenes. So in case of the oncogenes, first thing to remember is that there is only one mutation required. How you can remember this? O stands for oncogenes and 1 starts with O itself. So remember it this way, that there is one mutation required, and also there is the gain of function in case of the oncogenes. Initially the oncogenes are present in inactive case. The name for the same is proto-oncogenes because of the gain of function mutation in them in the any of the one of the allele. Reason being, they are dominant in nature. There occurs the conversion of the proto-oncogenes into the oncogenes. The second one is the TSG, that is the tumor suppressor genes. In this there are two mutations required. Remember it this way, tumor suppressor T and 2 also for T. So remember it this way, that in case of the tumor suppressor genes we require two mutations, and in this there occurs loss of function mutation. So depending upon the same, only these points are important and you will be able to solve many questions related to these topic. Reason being, many questions have been framed, so it's very important for you to have a good grip over this. I'll make a video for the questions as well, so do not worry about that. So this was all that I wanted to share in this particular video. I hope this particular video would help you out in some way or the other. If yes, do let me know in the comment sections below, and also do not forget to like, share it with your friends, and also subscribe to my channel, and also do not forget to join me on the Telegram channel as well. I have provided the link of the same in the description box. So thank you so much everyone for watching, and I'll see you soon. Bye.